Hello friends, today we are going to start a new topic, nucleic acid. First of all, discovery. It was first discovered by Friedrich Mischer. These are macromolecules. These are polynucleotides. They serve as blueprint. These have all the informations and are hereditary material. That is, these are passed from parents to the offsprings. Hence, these are important for continuity of life. Now, the types of nucleic acid. There are two types of nucleic acid, DNA and RNA. DNA. It is the genetic material of all living organisms. It is present in eukaryotes, in nucleus and also certain cell organelles like chloroplast and mitochondria. It is also present in prokaryotes. But here the DNA is not enclosed in a nucleus. Now the RNA, it is the other nucleic acid. There are different types of RNA, messenger RNA, ribosomal RNA, transfer RNA, microRNA, which have different functions. This we will discuss in detail in separate video. So nucleic acid is macromolecule. It has monomeric unit. This monomeric unit is nucleotide. Hence, nucleic acid is also known as polynucleotides. There are three subunits or components of nucleotides: a nitrogenous base, a pentose sugar, one or more phosphate groups. Let us see them one by one. First is nitrogenous base. Organic molecule which contain nitrogen. It is heterocyclic, planar, aromatic and water insoluble. It has amino group that can bind an extra hydrogen. Hence, it is base. So, it is called as nitrogenous base. There are two types of nitrogenous base, purines and pyrimidines. First is purines. This is the basic structure of purine. It has two rings which consist of carbon and nitrogen atom. There are two types of purines, adenine and guanine. This is the structure of adenine. Its chemical name is 6-amino-purine. This is the structure of guanine. Its chemical name is 6-oxy-2-amino-purine. Now pyrimidine. This is the basic structure of pyrimidine. There are three types of pyrimidine: cytosine, thymine and uracil. This is the structure of cytosine. Its chemical name is 2-oxy-4-amino-pyrimidine. This is the structure of thymine. Its chemical name is 5-methyl-2,4-dioxypyrimidine. This is the structure of uracil and its chemical name is 2,4-dioxypyrimidine. In DNA, adenine, guanine, cytosine and thymine is present, and in RNA, adenine, guanine, cytosine and uracil is present. So, what we see that in RNA, instead of thymine, uracil is present. Now, let us see pentose sugar, which has 5 carbon atom. These have D-stereoisomeric configuration. To differentiate the carbon atoms of sugar from the nitrogenous base, it is designated with prime, that is dash mark. In RNA, ribose sugar is present. This is the structure of ribose sugar. In DNA, deoxyribose sugar is present. This is the structure of deoxyribose. So, what's the difference between these two sugar? Here we refer it as deoxyribose because it has one missing oxygen atom at 2' carbon atom. If we see here oxygen atom is missing. Here oxygen atom is present. Due to the missing of this oxygen atom, it is referred to as deoxy. So now let us see nucleoside. When pentose sugar binds with nitrogenous base, it is known as nucleoside. Sugar binds with base and the bond between the two is beta glycosidic bond. This bond is between the carbon atom of sugar and nitrogen atom of the nitrogenous base. These are the structure of nucleosides. Pentose sugar is attached to nitrogenous base. In these two structure, the nitrogenous base is purine and here it is pyrimidine. These are the glycosidic bond. Nitrogenous base are able to freely rotate around glycosidic bond. Hence, they adapt different conformations. If we see these two structures, here the base is purine and due to its free rotation around the glycosidic bond, these adapt two different conformation. One is anti, one is syn. Here the base is pyrimidine. This is also able to freely rotate around this glycosidic bond, but it has only one conformation. Now let us see nucleotides. These are phosphoric acid esters of nucleosides. These consist of pentose sugar, nitrogenous base and phosphate group. And the bond between the pentose sugar and phosphate group is ester bond. So we can say that nucleotides are made up of sugar, base and phosphate. Since sugar and base combine and forms nucleoside, so we can say that nucleotides are made up of nucleoside plus phosphate. This is the pentose sugar. This is nitrogenous base and this is the phosphate group. And the bond between this pentose sugar and phosphate group is ester bond. This phosphate group is attached to the C5' carbon atom. So C5' carbon atom can have 1, 2 or 3 phosphate groups. And these are named as alpha, beta and gamma accordingly. These are monomeric units of nucleic acids like RNA and DNA. But they are also required for formation of energy currency like ATP and GTP. These are also precursors of coenzymes like NAD+, nicotinamide adenine dinucleotide, NADP+, nicotinamide adenine dinucleotide phosphate, FAD, flavin adenine dinucleotide and coenzyme A. Now let us see polynucleotides. Condensation of two or more nucleotides form polynucleotides. And the bond between two nucleotides is phosphodiester bond, which is a covalent bond. This bonding takes place between 5-hydroxyl group of one nucleotide and 3-hydroxyl group of second nucleotide. And in this process, water molecule is eliminated. These polynucleotides shows directionality, which is from 5' to 3' direction. So this is one nucleotide and this is the second nucleotide. And the bonding takes place between 5-hydroxyl group of one nucleotide and the 3-hydroxyl group of the other nucleotide. Water molecule is eliminated and phosphodiester bond is formed. This is dinucleotide. Similarly, further nucleotides are added and this forms polynucleotide strand. To understand it, we have represented the polynucleotide strand like this. Here, sugar, base and phosphate. This forms one nucleotide. This shows the direction 5' to 3'. So, new nucleotides are always added in the 3' end, that is in this end. This is actually the primary structure of the polynucleotide, and here the phosphate and sugar chain. These forms the backbone which runs along the length of the polynucleotide. These are the bases. Secondary structure is also formed, which is the three-dimensional structure of single polynucleotide strand or more than one strands. So if we talk about DNA, the three-dimensional structure is double helix and it has two strands that are antiparallel. One strand is in this direction and the other is in this direction. So if we consider two strands of the DNA, if this is the one strand which is in this direction, the other strand will be in this direction, and the pairing takes place between the nitrogenous bases. This involves hydrogen bonding between the nitrogenous bases. This is the three-dimensional structure of the DNA, which is double helix, where these two strands coil around each other. These are the phosphate sugar backbone, and these, the one with green color, are the phosphate sugar backbone, and the other, this red and blue color, these are the hydrogen bonding. But the pairing between the nitrogenous base is not random. There is certain rule known as Sharagraff's rule. According to this, adenine always pairs with thymine and it involves double bond. Guanine always pairs with cytosine and it involves triple bond. So, total number of purine is equal to total number of pyrimidine. But the percentage of cytosine and guanine is not equal to percentage of adenine and thymine. Also, amount of adenine residue is directly proportional to amount of thymine residue, and amount of guanine residue is directly proportional to cytosine residue. So this is all for today's video. In the next video, we will discuss about structure of DNA in detail. We will also compare different types of DNA. So stay tuned. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.